Dr. Jacqueline Rose, a new assistant professor at Western Washington, started in September of '08. What I work on is mostly the mechanisms of plasticity, of how our brain basically encodes and stores information over a long period of time. So basically the nuts and bolts of how our brain remembers things. Well, this is our second year running the Neuroscience on Tap series. So this will be my second time doing a Neuroscience on Tap. Basically, I feel like I'm hosting it. I present, my topic of research, the bigger picture, sort of how it affects individuals in the community, and then from there generate discussion that is more accessible to the everyday person. So we can talk about what we can extend from specific research to the larger community and say how their lives are affected by what we're learning in the lab. And the idea is through the repetition, it becomes sort of encoded into the circuitry.
I'm interested in how people create memories and how memories change over time. I guess the short-term gains I hope to get from this is maybe some trivial information I can share with my friends, or maybe some long-term information I can use when trying to understand how my memory is working later in life.
Questions. People are interested in, well, how is this going to affect me later in life? Or does this explain what I'm currently experiencing? Or does this explain memories I have from a long time ago? And we end up going through this sort of back and forth where one question will stimulate the next person to suggest something, and so on and so on. And so,
yes, you talked about the hippocampus getting smaller with enough stress. And what about that we build new neurons all the time in the brain? Where in the brain is that affected, or can that affect the hippocampus?
What do we know in the way of chemicals or events that are clearly demonstrated at this point to be to adversely affect memory?
I have a question about your worms. So, you said 302 neurons?
at last count.
Okay. How many does an average human have?
That's not about my worms.
Well, I just want an idea because I'm trying to figure out how big these worms are. How big are they?
They, in adulthood, they measure out to a millimeter in length. So they are. By participating in events where we're having to examine the big picture directly by talking to the community really grounds us in what the point of our own research is, like what's the end or what's the goal of our specific experiments supposed to address. It's a great advantage for us as well to make sure that we still have that perspective in our research. Thank you very much for coming.